Hello everyone, my name is Naval Singh and I am a final year PhD student at Loughborough University. In this talk, I will present our research work on the identification and characterization of new physical mechanism for reversible trapping of colloidal particles in dead-end microstructures such as these microgrooves via steady-state solute gradients in a continuous flow setting. The controlled transport of nanoparticles plays a key role in several technological applications, including drug delivery, diagnostics, enhanced oil recovery, or particle filtration and separation technologies. However, controlling the motion of nanoparticles within a confined environment such as porous media or dead-end channel can be very challenging. Indeed, flows cannot be used to advert the particles in or out of the pores, whereas the other obvious transport mechanism, such as Brownian diffusion, is slow and inefficient due to the slow diffusivity of nanoparticles. In recent years, a number of studies have addressed this challenge by exploiting the motion of sub-micron particles induced by solute concentration gradients, the so-called diffusiophoresis. In few words, diffusiophoresis causes the motion of a charged particle in an electrolyte solution when exposed to a gradient of salt concentration C. The resulting diffusophoretic particle velocity UDP is proportional to the gradient of log of C. The proportionality constant is DDP, which is diffusophoretic uh, coefficient, and is a contribution of two distinct phenomena, namely electrophoresis and chemophoresis. Externally applied unsteady state solute gradients have been adopted to boost the migration of nano and microchannels into and out of dead-end pores. However, due to the transient nature of these gradients, the particle manipulation capabilities are lost within a short period of time. Conversely, steady-state solute gradients have been successfully used to accumulate nanoparticles in target locations, but within open-ended channels such as T-junctions or at the inlet of glass capillary or within a nanochannel. Under steady-state conditions, the diffusophoretic particle velocity is counteracted by a hydrodynamic flow velocity u, and particle focusing occurs at the region where the total velocity, which is up, vanishes. However, for some micron particles, their slow Brownian diffusion cannot compete with the particle velocity, and under steady state gradients, the particle keeps accumulating at a point of zero velocity until the packing limit is reached, and this results in irreversibly clogging of the channel. In our study, we identified a new mechanism for rapid delivery and accumulation of sub-micron particles by means of steady-state gradient. The proposed mechanism is fully reversible and particles are trapped within a dead-end channel and can be transported in and out multiple times without irreversible effects such as clustering and device clogging. We fabricated a size-shaped microchannel by photolithography and soft lithography techniques. By means of a syringe pump, a lithium chloride solution at low concentration, which is Cl, containing fluorescent carboxylated polystyrene particles of 200 nanometer diameter were injected in the inner channel of the device, whereas the same solution without colloids is injected at the outer channel of the device. A flow switch valve is provided here to flow the switch between low concentration and high salt concentration, which is CH. And so in this case, 0.1 millimolar lithium chloride and 10 millimolar lithium chloride solutions were used to generate a steady state gradient within the device. A portion of channel is made out of optical glue NOA 81, the yellow region, which is glued on top of a silicon microgrooved substrate, the brown region. The grooves are perpendicular to the flow direction and hence act as a dead-end pores. A confocal microscope was used to perform a scan along the optic axis and determine the distribution of nanoparticles within the channel as well as within the microgrooves. The schematic represents the top and side view of the microchannel. The characteristic sizes of the groove and channel are shown. The groove and channel depth, which is small h and capital H, is about 50 microns, and the groove width T is 8 microns, whereas the groove pitch L is 32 microns. By pumping a low salt solution in the outer channels, no salt gradient is present and the particles remain in the main channel. And they do not enter the grooves, likely due to steric and electrokinetic wall exclusion effects. When the flow valve is switched and higher salt concentration solution CH is pumped into the side channel, particles rapidly accumulate at the entrance of the grooves as well as at the top wall. The top view shows the average particle concentration along the groove depth, whereas the side view represents the particle concentration in a plane parallel to the flow direction and perpendicular to the channel width direction. To quantify the particle trapping effect in the grooves, we can look at the particle concentration profile as a function of the channel and groove depth in the direction y. The origin of the depth axis y is taken at the top wall of the groove, and with the positive direction from the main channel to the groove. In the absence of the salt gradient, the normalized concentration profile, the blue curve, is homogeneous inside the channel and then vanishes inside the groove. When the salt concentration is present, that is the red curve, the two accumulation spots are observed, one near the top wall and one at the entrance of the groove. The intensity of this second peak is 3.5 times the bulk concentration. To quantify the trapping performance, we defined a trapping parameter A, which is the area below the normalized concentration profile in the groove region. The area A, 0.02 in the absence of the salt gradient, is raised to 0.8 when a salt concentration gradient is established. Here it is represented from the blue region to the red region. In other words, there is a 40-fold increase in the average particle concentration inside the grooves when a salt gradient is imposed. By tracking the trapping performance over time, it can be seen that after switching the valve, it takes only few minutes for a trapping performance to increase and it reaches a steady state. To understand the mechanism governing the particle trapping, we performed the steady-state numerical simulations in COMSOL. Briefly, the 3D simulation domain consists in a rectangular channel with a single groove located at a distance zg from the channel inlet. The steady-state Navier-Stokes equation is solved together with diffusion advection equation for the salt concentration C and the particle concentration N. The effect of diffuso-osmosis at the channel walls are also accounted for by introducing slip velocity u wall proportional to the diffuso-osmosis coefficient gamma do. The solution of the problem depend on the 5 a-dimensional numbers listed here. The details of the simulations are provided in our archive. The link is provided here, and I would also like to tell you that manuscript has been accepted on PRL, physical review letters. The particle concentration profile along the channel depth predicted by the simulation, which is the black curve, compares very well with the experimental curve, which is the red one. And a good agreement between the experiments and theory is also observed for the particle distribution on a plane perpendicular to the flow direction. The cross-section views shows here the salt gradient leads to the accumulation of particles just below the groove entrance, as well as the formation of two asymmetric and weaker focusing region close to the top flat wall. But the main question is why the particles get trapped at the groove entrance and not go deeper inside the groove. The salt concentration isolines are here shown in a plane perpendicular to the flow. In the inner flow region of the channel, these lines are bent towards the outer flow due to the Poiseuille-like velocity profile. This results in a Sol gradient and thus a diffusophoretic velocity, whose streamlines are shown by the white arrows directed from the channel bulk towards the flat wall at the top and groove at the bottom. However, if one observes the streamline of the total particle velocity fields, which is given by hydrodynamic velocity u and UdP, the diffusophoretic velocity, in a cross section perpendicular to the channel in the widthwise direction, which is the red shaded region, one realizes that as the particle migrate towards the groove by diffusophoresis, they are captured in the recirculation region just below the groove entrance and accumulate at the center of this region. Particles do not travel further down as diffusosmosis flow pushes the particle back to the recirculation zone. Importantly, our simulations show that in the absence of diffusosmosis, particle would still accumulate within the recirculation region, and in addition, they will also concentrate at the bottom end of the groove. Hence, it can be concluded that the observed particle trapping is due to the combined effect of diffusophoresis and hydrodynamic flow recirculation in the groove. Secondly, diffusosmosis is not required to produce the trapping effect. More interestingly, although the particle concentration peak is located where the in-plane zy particle velocity vanishes, the out of plane component of the particle velocity, which is the zx component, is not zero at the trapping point. Therefore, a vanishing particle velocity is neither a sufficient nor a necessary condition to achieve particle trapping and accumulation, as was assumed in the previous studies. As a result, in our system, particles are continuously transported in and out of the peak region, as shown by 3D streamlines of the particle velocity field in the groove. Finally, by using the switch valve, we are capable to alternatively remove and re-establish the salt concentration gradient and hence swap between concentration profile without and with salt contrast. As a result, the particle trapping performance can be repeatedly varied between 0 when there is no salt gradient to around 0.8 when there is a salt gradient, thereby demonstrating the full reversibility of the steady state diffusophoresis manipulation, which is achieved in our device. So in conclusion, we presented a new mechanism for particle delivery and accumulation within dead end structures under continuous flow setting and steady state solute gradients. We also demonstrated that particles can accumulate in a region of non-vanishing particle velocity, thereby preventing irreversible effects such as particle clustering and channel clogging. The average particle concentration in the focusing region was around 40 times the concentration of the particles in the inlet stream, and most importantly for applications, this phenomena is fully reversible. So particle can be cyclically trapped and released upon removal and addition of salt gradient. We believe that this new mechanism could unlock exciting opportunities for the exploitation of diffusophoretic transport for drug delivery, synthetic biology, and on-chip diagnostic applications, such as, for instance, the time-controlled sequential delivery of multiple drugs within dead-end pores, or pre-concentration of extracellular vesicles followed by sample release and off-chip downstream analysis. Finally, we would like to thank the funding agency that supported this research. Please check out our publication for detailed explanations. Many thanks for your attention.